Okay, well, good afternoon, good morning, or good evening, everyone, depending on where you are. Thank you for tuning in. I'll just go through a few housekeeping notes before we get started. Please remember to post any questions you have in the Q&A tab, and then I'll be able to get to them at the end of the talk and ask our speaker to answer those questions. Also, a reminder that if you want to catch past seminars, including today's seminar, see the website on the screen that you're seeing right now at the Connect Science website. And again, I would just like to thank Claudia Steele O'Sullivan for arranging this meeting. And with that, it is my distinct honor and pleasure to introduce a good friend and colleague of mine, Dr. Christine Chaffer, who comes to us from the Garvan Institute in Australia, which means that it's 4 a.m. there. So I definitely owe her lunch and a margarita or two. So thank you so much for being here at this time of morning for you. Dr. Chaffer did her PhD work in Melbourne, Australia, where she was really understanding the plasticity of disseminated epithelial cells, primarily in the context of breast cancer. And she brought her talents and skills to Bob Weinberg's lab at the Whitehead Institute as a postdoctoral fellow, where she really made seminal discoveries about breast cancer stem cell plasticity and means by which cells disseminate and become overt metastases. She published a number of landmark papers, among them one where she really understood the mechanistic basis for that plasticity. We're seeing the ZEB1 transcription factor poised at chromatin to unlock different programs in order to mediate that plasticity. And that work really launched her own career now as a principal investigator at the Garvan Institute in Australia, as well as a number of her trainees and colleagues. So with that, it's my distinct honor to introduce Dr. Chaffer. The screen is yours.
Thank you, Sandy, and thanks everyone for listening. Let's see if I can progress. Okay, so I just at the start will take 2 seconds to introduce my lab and thank them immensely for the amount of work that they have done and are doing currently in the lab. Today I'm going to present some news stories from particular Bayer, who's a postdoc in my lab, and I'll just go over some of our findings that led to kind of where we are today. So one of the things that we think about in the lab is this idea that cancer cells can reside in distinct phenotypic states. So we know that tumors are heterogeneous, and the cancer cells themselves are heterogeneous, and so is the stroma, the ECM, everything around it. So the focus of my talk and my work today is really going to be focused on specifically the cancer cells themselves. So why we think about this is because heterogeneity is really important. We know that it's associated with aggressive disease. We know that it is associated with resistance to chemotherapies, and this is overall leads to poor survival. So heterogeneity is really interesting. It's not a finite parameter. This means that it can vary between tumors. It can vary between tumor subtypes. And the other really challenging thing about heterogeneity is that it can change over time. And this means that it can vary with disease stage and whether we are talking about primary or recurrent tumors or metastasis. And so for this reason, I think that heterogeneity really is a major scientific and clinical challenge. It presents a moving target, and I'd like to show you some data today about why we think that this plasticity and heterogeneity has such an impact on therapies, on failure of therapies for aggressive and metastatic disease. So how do we get heterogeneity? Obviously, there's many different ways, and I'd just like to introduce these 3 concepts. So obviously, we're very familiar with this idea of clonal evolution, where the heterogeneity that we see arises from distinct clones that have distinct genetic mutations. What my work has been focused on is this idea that heterogeneity arises from plasticity. And this is where we have clones with all the same genetic background, but the heterogeneity that we see arises due to changes in epigenetics or transcriptional changes. And of course, it's also probable that we can have tumours arising that are actually a mix of both clonal and plastic heterogeneity. And this is obviously something that I don't think we're really able to clearly dissect yet, but I think it's going to be important in the future when we think about how we can treat patients. So in the clonal evolution model, when we treat with things like chemotherapy, I think what we commonly see is that whilst we have a mixture of clones that at the beginning, the chemotherapy itself can either select for pre-existing clones, but it can also create new clones. We see emergence of very minor clones that now have a dominant role at the metastatic site. With the plasticity model though, when we introduce therapies, for example, chemotherapy in, what we see is often an enrichment of a cancer stem cell or a dominant clone, and that once the therapy is withdrawn or the tumor regrows, we see heterogeneity that's recreated, and this is happening via epigenetic mechanisms. And the reason why I think this is important is because when we think about plasticity in this context, about molecular pathways that are being reactivated to create heterogeneity, we can think about understanding what these molecular programs are. These are then finite programs that can be predicted, and of course they can actually be targeted. So let's take a little bit of a look at cell plasticity in breast cancer. We commonly use these markers, so CD44, and there are others, CD24, CD104, to identify these cancer stem cells or tumor initiating cells that arise at the top of sit at the top of the hierarchy. And these CD44 high cells will differentiate into CD44 low cells that are generally poorly tumorigenic. So all of our metastatic and tumor initiation potential resides in this cell population. At the moment, it's our best understanding of how the plasticity is created between those CD44 high and CD44 low cells comes from our understanding of these developmental processes. So the epithelial to mesenchymal transition and the reverse process, the mesenchymal to epithelial transition. So what we've understand is that when cells undergo the EMT, it pushes them into a more mesenchymal state. And this process, this developmental process, has been co-opted in cancer. And when we think about that in the cancer setting, we see that these cancer stem cells tend to reside in this hybrid mesenchymal state. So they don't tend to push all the way to the end into a complete mesenchymal state, meaning that they still retain some epithelial characteristics. And it's these cells, when they move into this part of the EMT spectrum, have the most tumor initiating migratory capacity. And here are just some of the markers that we commonly use to identify cells at one end of the spectrum or the other. But what we actually think or are starting to understand about this plasticity program is that cancer cells really don't exist in one state or the other, that they're actually residing along this spectrum. And that spectrum of cancer cells and cancer cell phenotypes are sitting on a landscape. So we think about this adoption of the Waddington landscape, where we have wells that are stable states, and that cells can transition from one well to the other via these canals. And it's these processes where we see cells moving along the EMT or the reverse process, the MET, back from one state to the other, and that there might be other branches coming off here where we see that this more cancer stem cell state can actually move into a much more highly metastatic state. So this is what we think about when I think about the phenotypic plasticity that we're dealing with in the context of cancer. So in order to study some of the mechanisms that are contributing to cell plasticity, here's just two examples of some cell line models that we use. So this is basal or triple negative breast cancer cell lines, the HMLER and HCC38, which I'll be talking about a bit today. So when we use FACS analysis to stain these cells, you can clearly see that both populations, both cell lines have this clear CD44 high and a CD44 low population. And when we isolate out those cells and put them into 2D culture, we see that the cells have very distinct morphologies. So the CD44 low cells, very epithelial, and you can see this much more relaxed epithelial hybrid EM state within the 44 high populations. And of course, just to validate what we know, well, when we look at the EMT status of these cells, we see CD44 high cells express EMT transcription factors, for example, ZEB1, and low E-cadherin. And we also know that when we take these CD44 high cells, doing a tumor sphere assay, so suspending the cells in 3D and asking them to form a sphere, you can see that the 44 high cells retain all of this capacity to form spheres, and that the 44 low cells can't complete this sphere forming assay. So it's a really high throughput surrogate assay to start to look at some of these molecular programs. So I'd like to just talk a little bit about some data that we've published on plasticity in the context of breast cancer metastasis. And so this was a huge body of work that was extreme amount of fun that I did with, well, Sandy and I started when we were both in Bob's lab. And the culmination of this experiment was published in Nature Cell Biology and included the collection of 3 dogs and 4 children along the way. So what we wanted to understand was how metastases are forming, but we wanted to do this in the context of the primary tumor, because obviously when cells are initially metastasizing at those very early stages, there's still primary tumor present, and we thought that this might have an impact on the process. So what we did was to inject a primary tumor in the mouse, or we just injected PBS or matrigel control, so nothing. Then 2 weeks later, we injected the cell lines via the tail vein in the mouse. And we got this really interesting result. So when we inject the cells in the tail vein, when there's no primary tumor present, you can see that they beautifully form metastases in the lung. However, when there was a primary tumor present, we found that there was 0 metastases able to form in the presence of the primary tumor. So we did this in immunocompetent mice, and we saw the same thing in immunocompromised mice. So this was nude mice, and even more metastases in the nude mouse setting, completely ablated when the primary tumor was there. So we wanted to look at what was going on in those 2 different metastatic contexts. So here we're looking at tumors that, and we're staining for E-cadherin and ZEB1. So E-cad to mark the epithelial state, and ZEB1 to mark a more mesenchymal state. And these tumor cells here represented by green from large T. So when there's no primary tumor present, these cells that were CD44 high to start with, very rapidly, so this was within 2 weeks, have turned on E-cadherin and turned down ZEB1. So they've very quickly transitioned into that more epithelial state. However, when the primary tumor was there, we found that these CD44 high cells did not turn on E-cadherin. They maintained their high ZEB1 state, so their high CD44 state. And so these are quantitated here. So this was telling us in the presence of the primary tumor, these CD44 cells were unable to differentiate. So we asked the question, if we do the same experiment, but in the absence of any other primary tumor, and just say, let's overexpress ZEB1, so we'll force the cells to be unable to differentiate, what happens to their tumor initiating capacity? And so here we have CD44 high cells overexpressing ZEB1 that you can see in orange. And here are the same cells without ZEB1 overexpression. And you can see within 2 weeks, they've shut ZEB1 down. And here's the results with our tumor initiating experiment. You can see when cells maintain high ZEB1, they grow very poorly. So this was telling us that when cancer stem cells are unable to differentiate, they're unable to form robustly growing tumors. So I'm just going to quickly go over some of the actual mechanistic insight that we found about how these cancer stem cells are staying in that state. So what we did was look at the immune system infiltrates within these tumors in both our murine and human settings. And what we found in the murine models was that in the presence of the primary tumor, the infiltrating lungs here where the metastases were growing were filled with neutrophils, and this is quantitated below. In the human models, we've had similar results with the innate immune system, but different cell population. So when the primary tumor was present, and the cancer stem cell population is not growing well, we see that there's a massive increase in macrophages within the tumor. So this told us that when we have that primary tumor present and metastases that are unable to grow in the animal, the metastases are having an influx of the innate immune system. Now, to cut a long story short, we wanted to look at what was common between the neutrophils and macrophages. And with a whole lot of RNA-seq and PCR, we discovered that IL-1 beta was actually highly abundant in both the human and mouse settings in those metastases that were unable to grow. So we did this experiment where we said, okay, if it's IL-1 beta that's preventing these cells from differentiating, let's just give the CD44 high cells a dose of IL-1 beta and see what happens. So in this experiment, we took CD44 high cells and injected them with a PBS control, with a bolus of PBS. And as you can see, the tumors very rapidly upregulate E-cadherin and downregulate ZEB1. However, when we had IL-1 beta present, you can see now that the CD44 high cells do not upregulate E-cadherin. In fact, they maintain high ZEB1. So this was telling us that the IL-1 beta is preventing the CD44 high cells from differentiating, and that this IL-1 beta is mimicking that, having that presence of the primary tumor there. And to replicate what we see when the primary tumor was there, the addition of this IL-1 beta, you can see, also caused this very large influx of macrophages into the tumor. So I'm just going to summarize the rest of that paper because you can have a look at it here. But what we understand now is that CD44 high cells or cancer stem cells undergo differentiation to recreate heterogeneity when they're just growing on their own, so when there's no primary tumor present. However, we've found and shown in this paper that when you do have a primary tumor and you're looking at these very early stages of metastatic seeding, the CD44 high cells are unable to differentiate, so they maintain this ZEB1 high, E-cadherin low state, and this equals a very poorly growing, if growing at all, metastasis. And so what we showed, went on to show in this paper, is that the primary tumor itself is causing inflammation. And we found that there's an increase in myeloid cells within the bone marrow, and that this then leads to an increase in myeloid cells in the circulation. And as I mentioned, these myeloid cells in the circulation end up into an increased infiltration at the metastatic site, an increase in IL-1 beta at the metastatic site, and activation of the IL-1 receptor on the cancer cells, which prevents them from differentiating. So with that in mind, and this understanding that this plasticity, which is the ability of CD44 high cells to differentiate into an epithelial state, this is the mesenchymal to epithelial transition. And really very little is known about this process. So we have shown that we know that ZEB1 needs to be shut down in order for cells to undergo this MET. But beyond that regulation of EMT transcription factors or shutdown of the EMT transcription factors, we don't really know how this process works. And we needed to develop some easier model systems to start to look better at this MET process. And this is where the TumorSphere assay has come in extremely handy. So these CD44 high cells, when you culture them in 2D, in a mixed population, CD44 high cells are maintained in the population. So they don't, the 2D culture conditions favor that maintenance of the CD44 high state. And I think this is one of the reasons why we don't lose these stem cells or cancer stem cell-like populations from cultures, because the culture favors their maintenance in that state. But when we take the CD44 high cells and put them in the sphere and then digest these spheres and do FACS analysis, now we can see that the emergence of this epithelial CD44 low population. And this 3D assay is the only way that we've been able to get CD44 high cells to actually move back in vitro into this epithelial state. So it's actually been a really handy assay. So what we wanted to oops, doesn't really help much, does it? What we wanted to understand was to start to look at how the MET actually works. And so to do this, we decided to do this time course assay where we seed the tumour spheres and collect samples at over 30 days as the tumour spheres are developing. We then went on to do single-cell RNA-seq and also single-cell attack-seq on these different populations so that we could start to understand what these programs are that are driving that MET process. And to do this, this is work that we did in collaboration with Smita Krishnaswamy and her PhD student, Alex Tong. So Alex and Smita had developed this tool called TrajectoryNet, and it's published here at ICML. And what I'm going to say some words that explain how this works. Alex was able to use continuous normalizing flows and dynamic optimal transport to understand how to draw trajectories from one state to the other collected from our different time point studies across the tumour sphere assay. And so what we're looking at here is the projection of and the connection of how these, this is our single cell RNA-seq data looks in space. And so here you can see these are our tumour spheres at day 2, day 12, day 18, and day 30. And added in here, we're also looking at the population of this is HCC38 cells just in 2D. And one of the things we wanted to understand was what are actually the cells within this mixed population that are giving rise to the spheres. Now, we commonly use these markers CD44, 24, integrin beta-4, but they don't isolate purified populations of cancer stem cells, they merely enrich for them. So we really wanted to understand if we could define some better markers. And so here you can see, if we just look at this population of cells in 2D, that this marker, and here we're looking at EpCAM, you can see that EpCAM this is actually the CD44 low population, and you can see it highly expresses EpCAM. But you can also see within the CD44 high population that there are some cells that are expressing high levels of EpCAM, but the majority of the population doesn't. We also wanted to then look at other markers that would help us to better enrich for these cancer stem cells. And so what we found was that actually caveolin-1 was really highly expressed within a lot of cells within the CD44 high population. So with the help of Bayer in my lab, we did this FACS sort to isolate CD44 high cells from the HCC38 cell line. And then we first of all used these markers that came from Alex's analysis and looked at further sorting on EpCAM positive or EpCAM negative. So when we did this, what we found was actually enriching for the EpCAM positive cells didn't give us any significant enrichment over the unsorted population itself. But doing the EpCAM depletion did show that you could deplete for the cancer stem cells. So this was a nice way to deplete, but it didn't give us the ability to enrich for cancer stem cells. So then we did the same experiment, but this time isolating, sorting for caveolin-1 positive or caveolin-1 negative cells. And again, we weren't able to show that on its own, the caveolin-1 could significantly enrich or deplete for the cancer stem cell population. So then we combined these markers and did now a double selection for EpCAM positive and caveolin-1 or the depletion. And here you could see that we got now a really nice enrichment that when we use caveolin-1 plus EpCAM, that we can significantly enrich for cancer stem cell ability via the tumour sphere assay, and that depletion gives us a really nice depletion of cells that can't from the CD44 high population. So this gives us, just to show you that we can use this TrajectoryNet analysis to really gain a lot better insight into how we can better identify what the cancer stem cell population are. Another really interesting question that we asked with the help of TrajectoryNet and from our single cell RNA-seq analysis was this question about cell cycle. So we had also always wondered whether the state of the cell gives it some preference to be able to initiate the sphere in the first place. And so this was an analysis that, that Alex did using gene sets that mark G1, G2, M, or S. And this is the 44 high population, and this space means something in this projection. So the cells that are closest here to the next time point are the ones that are where the trajectories will be drawn. And so this analysis actually told us that these G2 cells were the cells most likely to be moving on to the next time point and giving rise to the sphere. So we decided to test that hypothesis, which was pretty easy to do. We've injected our cell line with the Fuji cell system so that we could sort for G1 cells in red, S phase in orange, or the G2M in green. So we did this, and then by FACS analysis, just put these populations into the tumor sphere assay. And to our surprise, because I was predicting that it would be the G1 cells that would give rise to spheres, we were able to validate Alex's predictions that actually these G2 cells really are highly enriched for that capacity to be able to form a sphere. So we're really, really excited by that finding. Another important question that we wanted to ask with this trajectory net analysis is to understand what's going on at day 28. So within those spheres, I showed you that when we look at the later time points by FACS analysis, we know that these epithelial populations are rising. And so here we're looking just at the day 28 time point, and we looked at the different clusters of cells. And so we have these cluster 0, cluster 1, and this is actually apoptotic cells also at day 28. And using this tool that was developed by Mohit Joly in India, we can give the cells a score, an epithelial mesenchymal score. So this is published in this paper. And so what we found that in this cluster that was appearing as 0, that it had a higher mesenchymal score, whereas this cluster that was appearing down below had a much higher epithelial score according to this scale. So now we can look at our single cell data and ask what factors are either enriched in this epithelial population and what factors are enriched in this mesenchymal and are actually getting turned off in this epithelial fraction. So we're currently looking at this analysis at the moment, and we should have some new data from this soon, but we've been able to identify factors that we think might actually be pushing these cells down into the epithelial state, which is really exciting. So TrajectoryNet has been a really amazing tool. We've been able to identify markers that we can better enrich for cancer stem cells from populations. And we've also been able to understand what factors are going on at these latter time points. So what's driving MET within the populations. And currently we're still working on, I think the most exciting part, which is actually looking at the flux of transcriptional programs that occur throughout the trajectory. And with this analysis, we hope soon to be able to really define different programs that are going on a single cell basis that are driving these trajectories to the more epithelial state and more mesenchymal state. So I'd just like to show you, really just to introduce some other experiments that we're doing in this metastatic setting with respect to cell plasticity. And these are studies that were led by Bayer in my lab and Sarah Kummerfeld and Scott Yulton from the Garvin. We wanted to ask the question about what is going on with metastases at different sites within the body. And so to mimic this, what we did, we used the MD8231 triple negative breast cancer cell line, injected it into the memory fat pad. And then we allowed metastases to develop throughout the mouth. And then we harvested primary tumor and sent that for single cell sequencing. And then we also, this was a survival surgery. So we pulled the primary tumor out and then waited another 4 weeks for the metastases in other sites of the mouth to grow bigger so that we could also single cell seq those. And so what we collected was primary tumors, lymph node, liver mets, and lung mets. And so what we've been trying to do is understand what these transcriptional programs are that are going on at these different sites. within the mouse. And so here's just the preliminary analysis that we've been doing. When Scott combines all of the single cell data from these different sites, this is looking at all of the sites here, we can see that the cells form into about 9 different clusters. And so the really interesting thing, if we just look at this picture here, is that when we look at the clusters of cells that arise, that different cell types are present at, represented at different, ratios within different metastatic sites. So here you can see that there's representation of almost all 9 clusters within the primary tumor, but we can see that the lung looks markedly different to the liver and the lymph node. It's really dominated by cluster 0, whereas we see that these transcriptomic programs are very different in the liver and also in the lymph node. So we're currently working through these data at the moment, but there's some really exciting trajectories about plasticity going on within these different metastatic sites. Another thing that we're working on is in collaboration with Joachim Lundeberg at Karolinska and Annelie Molbrink, who's done some spatial transcriptomic analysis for us. And what we've done is taken the same tumors from those mice, and we've taken slices. And so we've been looking at the spatial transcriptomic profiles of primary tumors versus the lung. So here you can see the MET in the lung, and here's a MET in the liver. And here we're looking at the representation of, so this is human genes, so you can see the MET, and here's the human genes in the liver. And this is the same for the lung and same for the matching primary tumor. And with work still in progress, we're working with SMITA also to be able to align the single cell RNA-seq data that we have with this spatial, to overlap that and align it with this spatial transcriptomic data. So there's some really, really interesting work going on in that space. I'd like to just focus now on the reverse process, the EMT, and I'll just start with some of the work that got us excited about this EMT process in the first place. And so this was work that I did do during my postdoc, and it was really trying to address this understanding of cancer stem cells versus non-cancer stem cells, so CD44 high versus CD44 low cells. And it really came from this experiment where we were trying to show that non-cancer stem cells are non-cancer stem cells, they can't initiate tumors. So what we did was to purify these CD44 low cells and inject them into mice. And what we found was, in fact, CD44 low cells are quite capable of forming tumors. And so this was surprising, but we started to wonder either the cancer stem cell hypothesis is not true or there's something different going on here. So what we did was to take these tumors and digest them and relook at FACS by FACS analysis. And what we found in all of these tumors was that the 44 low cells had recreated a CD44 high population. And here this is the quantification of that. So this tells us that CD44 lows can initiate tumors, but ask the question, can they only initiate because they're able to recreate that CD44 high population? So in order to get at that question, we needed to understand how we could block the ability of these CD44 low cells to move back into this cancer stem cell state. And obviously we know that the EMT transcription factors drive the EMT. So the first place we looked was, are any of these EMT factors upregulated? And this is where we found that ZEB1 was the predominant transcription factor that was increasing from CD44 low to CD44 high cells. So we then did this same experiment again, but this time we injected CD44 low cells or CD44 low cells expressing shRNA to ZEB1. And now when we did this and blocked the ability of these cells to be plastic, now we could see that CD44 low cells could not initiate tumors. So this really led us to the conclusion that that plasticity is really important for tumor initiation or for non-stem cells to be able to tumor initiate. And what we wanted to understand next was where is this actually happening in the cancer setting? So we decided to look at in this idea that the cell lines that we've been working with were all on the basal triple negative side. And so we asked the question, is this something that all 44 low cells can do, or is it more restricted? And what we actually found was that when we purify 44 low cells from luminal cell lines and measure their ability to be plastic, spontaneously plastic in vitro, that we don't see any movement at all within the luminal breast cancer cell lines. However, when we looked at basal cell lines, we found that they all had the capacity to be able to spontaneously switch back into that CD44 high state. So this led us to look at the epigenetic status of ZEB1 because we know that ZEB1 was important for that plasticity. So we looked at the ZEB1 promoter, and when we looked within the CD44 low cells, we found that the ZEB1 promoter had evidence of both K27 trimethylation, but also K4 trimethylation. So this was telling us that CD44 low cells are maintaining the ZEB1 promoter in a bivalent chromatin configuration. When we looked at the CD44 high cells, you can see where ZEB1's active. Now we see that the K27 trimethyl is resolved and that they only have the active K4 trimethyl mark. When we looked within the luminal fraction, now we saw again something really interesting. These cells that are not plastic, that are really unable to spontaneously switch from 44 low to 44 high, were really strongly marked by K27 trimethyl without the K4 trimethyl. So we have this idea that these cells that are less plastic or less able to move spontaneously have this ZEB1 promoter in this repressed state, and that this ZEB1 bivalent chromatin configuration is really identifying those cells that have that propensity to be plastic. Now, I'm moving on to some newer data here, following on from that idea, and it's coming back to this concept that I was mentioning at the start, the idea that chemotherapy itself can drive plasticity. So we and others have looked at this ability of chemotherapy to drive phenotypic plasticity. And here's some data out of our lab. And this was looking at the ability of these common standard of care chemotherapies for triple negative breast cancer. And what we were consistently able to show is that when you have CD44 low cells and treat them with increasing doses of chemotherapy, that chemotherapy pushes these cells up into the CD44 high state. And so when we think about this on the cancer cell state landscape, what we're seeing is that for some reason, this chemotherapy is able to really push these cells up into this state and prevent this differentiation from occurring. So in order to be able to translate this into a therapeutic strategy, we needed to understand beyond ZEB1 what might actually be druggable and thus targetable as a therapy to prevent this type of plasticity from occurring in the presence of chemotherapy. So in order to answer this question, Bayer isolated 44 low and 44 high cells, and these were from breast cancer cell lines from the triple negatives. And we did RNA-seq, ChIP-seq, and have been putting this work together to try and understand the plasticity network. And so here, these analyses were done by Melissa Davis from WeHi in Melbourne and her now PhD student, Saror Hediezadeh. And we were asking the questions, what's the difference between 44 low and 44 high across these cell lines? And what we first did was to look at how similar these breast cancer cell lines were or are to patient samples. And to do this, Saror took all of the TCGA samples and projected them, so these are marked by the crosses, in this UMAP space. And And what we found, and then, so these are the crosses, and then the circles represent the breast cancer cell lines that we projected on top of the TCGA. And so what we're able to show here is that our luminal, so these are MCF7 ZR75-1 cells, overlap with the luminal phenotype within the TCGA breast cancer samples. And here we can also see that when we project our CD44 high and CD44 low cells into the same space, you can see that they also overlap with distinct populations of triple negative breast cancer cells, or of the basal population of cells. So this analysis told us that the signatures of the cells that we're looking at are evident within the TCGA samples. We also then pulled out those TCGA samples and looked at survival analysis, and you can see that when we pull out the TCGA samples that are similar to our CD44 high cells, you can see, even though this is not significant, I think due to the low sample number, but you can see that there's a clear trend towards the CD44 high-like tumors to be doing worse compared to the rest of the basal tumors. Now, triple negative can be divided into 4 molecular subtypes. This work was published by Lehman et al. And so basal-like 1, basal-like 2, mesenchymal, and LAR. And we wanted to look at where our breast cancer cell line subpopulations projected into that space. So this analysis again was done by Sarora and Mel. And what we know and found is that the HCC38, according to the Lehman phenotype, classifies as BL1, BL2. So same for HMLER. When we look at the CD44 high subpopulation within those cell lines, now we see that those CD44 high cells are most similar to the mesenchymal subtype. So this is just really to point out that the molecular programs that are driving the CD44 high or this cancer stem-like state are really quite distinct from the bulk of that non-cancer stem cell population. And this is really just to further iterate that, bulk analyses don't really capture the molecular programs that are driving the more aggressive subpopulations within the tumor. So we then wanted to identify factors that would be from our analyses that are driving plasticity. So we looked for genes that were upregulated in CD44 high across the cell lines. And so we did a combination of different analyses. So these were discriminative analyses and correlative analyses. And we also then put on 2 additional factors. So we wanted to look for a transcription factor, but that's not ZEB1. And we also had another criteria that whatever we pulled out of this analysis had to have known drugs available so that we could start to treat them animals to block plasticity. And sorry that I can't divulge the targets just for this part due to patents at the moment. But what we were able to show here is that previously I'd showed you that when you treat CD44 low cells with chemotherapy, it pushes them into the CD44 high state. So this is red here, and here we're treating with doxorubicin. When we use an antagonist to our drug target, to our gene target, we were now able to show that in the presence of chemotherapy, this drug is now able to prevent these CD44 low cells from moving up into the CD44 high state due to the chemotherapy. So we're really excited by these findings, showing that we've actually got a way to stop plasticity that's driven by the chemotherapy from occurring. So we then wanted to test these analyses, test this drug in an in vivo context. So here, what I'm showing you is the 231 breast cancer model. And so here's how the tumors grow on their own. The purple is docetaxel chemotherapy on its own. And we have, there were 3 known drugs to this target, so we tested all of them. And what we found was that 2 of these drugs, when combined with chemotherapy, were, and this gray is the first cycle of the drug treatment. And so the gray is the drug treatment on the whole time, and here's weekly doses of docetaxel. And when the grey bar ends, this is a drug holiday, and then we did a second cycle of treatment. And so what you can see is the tumours start to respond to docetaxel on its own. They continue to decline a little bit, and then we see that at the second round of docetaxel treatment, these tumours are already resistant. And we found that 2 of these drugs had no effect when we combined them with docetaxel. But we did have this really exciting finding by one of the drugs, showing that we got this beautiful sustained inhibition of tumour growth when it was combined. So we were able to show that this drug completely sensitizes these tumours to docetaxel. And here's the survival outcome. We've then also repeated these studies now with multiple different PDX models, and here's just an example. So again, this is 2 triple-negative breast cancer models, and you can see very similar kinetics. This one is quite a slow-growing PDX. The purple is chemotherapy, and you can see that when we add our drug in with the chemotherapy in red, that we get this beautiful change in the kinetics and inhibition of tumour growth. With this model, PDX, similar model with a bit faster growing, we've been able to do multiple rounds of treatment. And again, we see similar things, that docetaxel works on its own, but by the second round, we already see resistant disease emerging. However, when we combine our drug, you can see the completely different growth kinetics, that the combination is really inhibiting the growth of the tumour. So based on these studies, we're about to launch a clinical trial in the metastatic breast cancer setting to test this combination. So I'd just like to finish by re-highlighting how we think the cell plasticity landscape looks. So cells are residing in a spectrum of states within this cancer landscape. And whilst I haven't talked about that landscape today, definitely fibroblast, immune system, the matrix is all having an impact on the ability of these cells to move across the landscape. But what we're really focusing on, what's inherent in the cancer cells themselves and what enables them to actually move between these different wells. And so we've shown in our lab and others that chemotherapy pushes these cells epigenetically into this more chemotherapy resistant state or cancer stem cell state. And excitingly, now we've thinking about these state gating therapies, where if we can prevent plasticity from occurring, that we can get tumours to respond much better to chemotherapy because they're unable to move into that chemo-resistant state. So there are multiple ways that we can think about doing that. We can think about treatments that push, that drive the MET, so push cancer stem cells back into an epithelial and more resistant state. We can also think about strategies that block the ability of these cells to undergo the EMT in response to chemotherapy. And of course, we can think about blocking both the EMT and the MET at the same time, and that by pushing cells back into this well, we can really get a much more enhanced response to chemotherapy. And so with that, I'd just like to thank all of the amazing people that I've been able to work with. I'd like to thank the Garvin, who's been supporting me in my lab since we came back 2018. I think I've mentioned the collaborators throughout, so just a huge thank you to everyone, and thank you for listening.
Great, thank you so much, Cece. That was a beautiful talk. It's really nice to see what kinds of insights you've gained. We have a number of questions that are still rolling in, so let's get right to those. So one question is, have you tried to put these cell lines through multiple cycles of EMT, MET, and tested their phenotypes?
No, we haven't done multiple rounds. Yeah, that'd be interesting to see whether you actually get to a state where cells become irreversible. Yeah, and I think you possibly do get there. We do see evidence, especially within the cell lines, that you can, if cells get pushed to that extreme mesenchymal state, that it's not as easy to get back once they get locked in that mesenchymal state. So I definitely think that this hybrid state is is semi-stable in the middle, the cancer stem cell state. And we see cells going either direction to 44 low epithelial or more mesenchymal. And of course, I guess the other example is that, what we see with the luminal cell lines, that those cell lines really are a lot more further down that epithelial state, and they definitely are much harder to move back up. You can do it if you overexpress transcription factors, but if we think about spontaneous plasticity, they don't naturally have that ability to move as far. So I think that the further along those spectrums you get, the harder it is to get out of them.
Yeah, that makes a lot of sense. It also makes me wonder too if your efforts to really define the cancer stem cell state, from your single cell analysis, et cetera, will lead to that understanding as well. Okay, some more questions. There are a lot of questions about your drug target, obviously, and I know you told us that due to patent issues, you can't really tell us, but I wonder, like, from some of these questions, if you can touch on it. One question from Lee Ellis is your combination effect inducing an immune response?
So I guess this is one of the limitations of doing these human PDX models, that we haven't really been able to look too much at the immune system yet. We've done combination stains looking at PD-L1 and things like that in TMAs to try and get an understanding of what might be going on, but it's one of the correlative studies that we're going to be doing in the clinical trial, like what's actually going on with the immune system when we treat in this way. And I think probably the key thing, and this is stuff that's been published, is that when you look at the immune system in the context of an epithelial tumor, that you versus the more mesenchymal type tumor, that we definitely know that those mesenchymal tumors are a lot colder with respect to the immune system. And so kind of what we're hoping is that once we push those cells with this therapy into this more epithelial state, so that they're more responsive to the chemotherapy, an added benefit of that might be that we actually change the immune environment also into a state that's really more cytotoxic. So whilst we don't have any answers there, we're hopeful that it might push in the right direction.
Yeah. And that, sorry to interject my own question there, but along those lines, it makes me wonder, because in vitro you showed that your drugs were very efficient at keeping them in the non-stem cell state, but it didn't seem, it's hard to tell, but it didn't seem like you had a lot of cell death, whereas in vivo you were getting that regression. So a beautiful, understanding. I think you're poised to address there. I just want to remind everyone, please post your questions in the Q&A tab and not the chat tab, so that I can be sure to get to them. Another question is chemo-induced EMT reversible upon drug removal?
So from, I don't know if I have an answer to that, because when we do these studies in vitro, the cells, chemo-reduced, so in vitro, when you push cells into the 44 high state in vitro, they stay there. So the only way, even if we withdraw the chemo, the natural state is once you've pushed into CD44 high state in 2D, you stay there. So the only way to see whether it would be reversible, which is an experiment we haven't done, would be to then lift those cells and put them in vitro into a tumor sphere assay, because it's the only way to actually make them differentiate again that I know of. Which, that would actually be a really interesting experiment. We haven't done that one.
Another question, are those drugs specific for docetaxel or any chemo-induced plasticity?
No, so we've, in vitro, we've tested a lot more drugs. So we've done cisplatin, doxorubicin, docetaxel. In vivo, with the intent that maybe this could be something that could combine with immunotherapy, we've done paclitaxel and nab-paclitaxel. So depending, they both work to varying extents, but also what we're seeing, which is interesting too, that within the PDXs, we see that they've got the PDXs themselves respond differently to those chemotherapies. So some of those chemos don't actually work themselves on the PDXs. So cisplatin's a good example; it didn't work. Nab-paclitaxel, we only got mild inhibition of tumor growth with nab-paclitaxel, and then mild further but significant due to the addition of the drug. So I don't think we've tested enough models, but definitely there's evidence that this is not something that's specific to the chemotherapy, that we'll be able to combine it with others.
Mm-hmm. Makes sense. Another question touching upon your drug target. The target for inhibiting plasticity that you identified, can you say if it has an effect on the epigenetic state?
We haven't looked. To be honest. But yeah, that would be interesting to look at as well. Yeah. Mm-hmm. Sorry, I don't have an answer. It'd be a great experiment.
Have you considered using CRISPR screens to identify genes that prevent transition of 44 high to low following chemotherapy treatment?
Yeah, we have. And part of that is because we're just setting up a CRISPR facility here, so a screening facility. But it's something we've talked about, and I think it's a great experiment too. Yeah, for sure.
Cool. A question from Patrick Anderson. Interesting talk. Regarding drug A, you mentioned that the trial will initially look at metastatic breast cancer. Have you looked at the effect of drug A on established metastases in your mouse models?
Yeah, sorry, I did, and I thought I was going to be running really short on time, so I didn't I didn't put in the metastasis data. But yeah, so what we've done in vivo is to so this definitely affects metastasis as well. So there's significant reduction in metastasis with our drug. And in fact, one of those drugs has the reverse effect, even though it's got the same target. So a slightly different mechanism of action. So there's some insights that we're looking at there. But in one case, we actually see an increase in metastasis with one of those drugs, which is like super interesting. So definitely our drug is inhibiting primary tumor and metastasis as well. So the metastasis survival data is also really significant compared to the docetaxel alone. So yeah, we are starting in the metastatic setting with some toxicity studies to do this combination treatment because it hasn't been done before. And then, you know, ideally, where we see these kind of therapies hopefully fitting in the long term is actually in the neoadjuvant setting. So what we imagine is that, and we've actually got data which I didn't present on this, but when we look at the drug target and look at patients, so we have access to a cohort of clinical samples where we have pre, mid, and post chemotherapy. And what we find is that the drug target actually acts as a biomarker. And if the patients have this biomarker from the outset, we can predict that they're the ones that are going to respond poorly to chemotherapy. And so when we think about this in the context of triple negative, we know that, you know, there's about 70% of patients respond really well to chemotherapy and get a pathological complete response. But then there's the other 20-30% of patients that don't get a pathological complete response, and they're the ones that go on to will always get recurrent or metastatic disease. So ultimately, what we're thinking with these plasticity or state gating therapies is if you can put that in the neoadjuvant setting, then we can get all patients responding to first-line chemotherapy, to primary chemotherapy, and not get that recurrent metastatic disease. So that's kind of our longer-term goal.
Yeah, that'd be phenomenal. And kind of along those lines, one of the next question from Maria Caramari, wonderful talk, excellent data. Bearing in mind that cancer plasticity and metastatic adaptation are highly dynamic procedures, how would you define cell autonomous versus non-cell autonomous mechanisms of different trajectories?
Yeah, I think that so this is kind of where we're heading with the spatial, to try and understand, you know, because we can't define these different cell states so clearly yet, and we have, you know, have had such poor markers, it's really, we don't understand how that architecture works within a tumor. Where is this cancer stem cell niche? What are they residing near? What other interactions are actually driving and maintaining that state? So we're hoping to get at that with some of these spatial and overlaying our single cell with the spatial data to get a much better understanding of the context and signaling context. I guess in answer to your question, because we've been focusing on what's actually going on in the cancer cell, irrespective of, you know, where the signals are coming from, is kind of what I'm thinking at the moment. So, you know, the work we did with Sandy was looking at what contextual signals were driving, maintaining that state. And there we could clearly show that it was coming from the immune cells. that they were impacting which cell state they were staying in. But we were still able to knock out the IL-1 receptor on the cancer cells and show that irrespective where the IL-1 beta was coming from, by affecting the cancer cells, we were able to push them in or out of that CD44 high state. So I think it's all going to be really interesting to understand from the context of also being able to, in the future, address which patients have these cancer stem cell-like populations, or which patients have that are epigenetically poised to use plasticity in this manner as a mechanism to avoid chemotherapy, or as a mechanism to avoid any targeted therapy. So long-winded answer, but I think it's important. But by focusing on the cancer cells at the moment, I think this strategy has led us to a therapy, let's just say, to start, or potential therapy. Yeah.
Actually, yeah, that your answer provides a great transition into a couple of questions that I'll combine here. First of all, did IL-1 receptor expression track with your E and M score states that you've defined nicely with your collaborator? And another question, have you considered combining IL-1 beta or IL-1 receptor inhibitor treatment or blockade with chemotherapy?
We have, and we have, and so some of those experiments, so we had initially started with anakinra, and then we didn't get any drug supply from the drug company anymore. So we actually put those experiments on hold, but absolutely, we're definitely going to do those experiments. And, there are some other transcriptional targets too that I think could also be, with known targets that might actually make a lot of sense that could actually be affecting plasticity as well. And I think that even within these cells, so I think that there's a lot of that context, like they have the potential to respond to multiple different stimuli. And this is why we're looking at this, have done that experiment where we look at the same cells but in different metastatic organs, because I think this is like probably the best example where you have, a really kind of, compared to a tumor, potentially homogeneous cell line in the 231s, but when you look at them, the transcriptomic profile, so the plastic adaptation to those metastatic sites is markedly different. And so I think that really speaks to how these cells can use a lot of different mechanisms at the same time in different sites in the animal. And so what we're hoping to do with that experiment is to take this concept, what we've done at the moment with this drug chemo combination, and ask that question, do we need organ-specific plasticity therapies to target liver versus lung metastasis? And based on the transcriptomic profile, I'd almost have to argue, yes, they just look so different. So I think that's a really interesting question, and something we're really excited to look at too. Yeah.
Maybe time for one more question, and then please don't worry. The rest of the questions will be sent to you by Claudia, so you can answer them in your own time. Have you identified cancer stem cells in human tumor tissues? Are there treatments available currently for patients who have cancer stem cells? Is a combination of chemo and a cancer stem cell inhibitor possible? I think
so no, I think that's what we're aiming at here. So no, to my knowledge, there aren't any therapies that target cancer stem cells at all, because we just know so little about what maintains that state. Mm-hmm. The therapy that we're looking at does two things. Because the gene's upregulated in the cancer stem cell state, even though I didn't touch on it today, we've actually shown that the drug inhibits cancer stem cell ability. So we've done this in the tumor sphere assay. So when we treat with this drug, it inhibits the ability of the cells to form the tumor sphere. And when we look at the CD44 low population, it inhibits plasticity. It inhibits, because they can't turn it on, it prevents the cells from moving. So we actually think that this therapy in particular is doing both of those things. So targeting the existing cancer stem cells and preventing the chemotherapy from enabling new ones to arrive.
Excellent. Well, unfortunately, our time is short, but thank you for leaving sufficient time for discussion. We have a number of questions still and a number of great suggestions in the Q&A tab, so we will send those to you for sure. But again, thank you, Dr. Chaffer, for your time and especially getting up so early, driving into work at 3 a.m. Again, I owe you a margarita or two.
I'll take the two snack.
And thank you, everyone, for tuning in.
Thank you for the questions and suggestions, and please reach out if I haven't answered anything you'd like more detail. I'd be very happy to answer. So thank you.
Thank you. Bye.